I bet a bunch of alums come back and they say, man, it's, you know, it's strange. I never thought I'd be here. And I never thought I'd be here. And it's mostly because I hated school, like forever. Undergrad, master's, I don't like authority and top-down control. I don't like regiment. It's driven me crazy. But I had to eventually go back and do a PhD because I love research, and it had to happen. So the stuff I'm going to present to you today is a summary of my dissertation work up at the School of Aquatic and Fishery Sciences at University of Washington. A dissertation which took me 4 years less than my master's and was 236 pages less, even though I got cancer up there and was sick for 2 years. So in any case, my talk today, I'm going to start with just a background about skates. Unfortunately, you people have been subjected probably to more skate talks than been given in this room than in any room in the world over the last 10 years. So whereas these are usually creatures that people don't generally know a lot about, you're probably more informed than most. But I'm going to start with a background on skates, what they are, what they do, why we care about them. My talk's broken into two main sections, kind of an ecology of Eastern North Pacific skates, which is broken into spatial association and trophic relationships. That's what I'm referring to as resource utilization. And then an applications for management section, which we'll finish up with. So the batoids are a monophyletic sister group to sharks. They contain the skates. About half of all batoids are skates, and they make up about a quarter of all chondrichthyan diversity, which is pretty crazy when you think about it. Most of what's been said about skates is one of the reasons is because they're difficult to identify, they're morphologically conservative, is they've been kind of grouped over time into the thinking that they're generalist predators on unconsolidated substrate, and which leads you to the idea that perhaps, or the kind of overview that they're somewhat ecologically redundant. They've been grouped for management purposes and for scientific studies because of that. This is one of the things I kind of wanted to look at, was the ecology of this group a little bit more, because it didn't make sense to me that there'd be so many skates if they're all doing the same thing. So biology of skates, like I mentioned, they're morphologically conservative, makes them hard to identify. Elasmobranchs in general have relatively conservative life history characteristics. This is true of skates, especially those large species and most species in deeper water. Not all of them. There are some small nearshore species that are relatively fecund and quick growing. They're one of the most widespread of all the fish groups. They occur from the intertidal down to over 4,200 meters with tropical submergence, but from the tropics to the poles. And like I said, they've just been considered to be generalist predators that live largely on unconsolidated substrates. So there hasn't been a whole lot of management or scientific attention paid to skates over time. It's actually been changing in the last 10 or 15 years. One of the reasons is because they've been recognized as they're abundant meso and upper trophic level predators, and there's this idea that they may compete with ground fishes for food resources has been looked at in the northeast coast with regards to the cod collapse for a long time, as to whether when some teleosts get overfished, there might be niche replacement with skates. One of the things that's starting to drive more of an interest in them. There's also fisheries. They've also been affected by exploitation, mostly in indirect fisheries, especially in the northern Atlantic and a variety of other regions where fishing pressure has been intense, like off South Africa as well. And it's altered assemblage structure in areas where they have long-term data, and largely the large, slow-growing species tend to get replaced by the smaller, quicker-growing ones. And there's been especially large declines in large nearshore species like this big skate, which we really don't know very much about. And this has been a species that now is of kind of growing concern to National Marine Fisheries Service on the West Coast, because we haven't seen, they're supposed to approach lengths over 2 meters, but we haven't seen one over about 1.4 meters in a long time, and their distribution is kind of concentrated near shore, and there's this idea that they may have been historically overfished. So for a lot of these skates, where we have no landings history going back, it's very difficult to monitor, to figure out what the population's doing right now, because we have no baseline. One of the methods that people are starting to look at, and one of the things we're looking at, is doing catch reconstructions and using known associations, spatial associations, and associations with other fishes that co-occur in landings. And the results of this study are going to go back, hopefully, to feed into some of these things for stock assessment. So let me introduce you to the skates I'm going to be talking about. I have two assemblages of interest. They're all specimens and samples that I collected, or we collected while we were at the Shark Center here over the course of time. And from the West Coast assemblage, we've got the big skate and longnose skate that both occur in the Gulf of Alaska assemblage too, which I'm going to be talking to you about in a minute. They're the two largest species. Big skate grows to just over a meter, and the longnose is just under a meter. Then on this coast, you've got the sandpaper skate, California skate, Scary skate, all species that grow to maximums between about 65 and 75 centimeters. The numbers you see there are the numbers I got from my spatial analysis on the left, and the numbers of stomach samples with full contents that I use for the diet analysis. Gulf of Alaska, like I said, we've got two of the same species, and then we've got this other suite of species. The interesting thing about the other suite of species there is they're considerably larger. These are assemblages when I say Gulf and West Coast assemblages. These are assemblages out on the shelf and upper slope from about 100 meters down to about 600 meters is where in both regions, these 5 species make up about 95 to 90% of the skates that you'll find in those regions. In any case, like I was saying, all these skates up in Alaska, the secondary species, the ones that aren't, well, not the secondary species, but the species that don't co-occur in both regions, these other skates are all grow to over a meter. So you've got basically an assemblage of larger skates on the shelf and upper slope in Alaska and smaller ones in the West Coast. So breaking my general questions down, these are kind of overall questions that I was asking, and then I'll break it down into the specific sections. But wanted to see if there was differential core resource use among sympatric skates, kind of going with a traditional niche approach. If there's competition for limited resources, I want to look at the two main aspects of the niche, space and food, to see what they might be, where there might be overlap or separation. And then, like I mentioned, what's the relevant importance of these two factors. And then for fisheries management, I wanted to use the results of the ecological work to improve management of skates, mostly through emerging ecosystems approaches, which in Alaska, which I'll mention in a little while, mostly consist of trophic or food web modeling. On the West Coast for the ground fishes, it's largely spatial. Okay, so first part in spatial associations. The first, I'm referring to West Coast skates because I've got 3 kind of components to the West Coast skates: the spatial, trophic, and the fisheries management. And some of them encompass the whole coast. Some of them are just based off this kind of central California region. So I'll show you the footprints in a little bit. Off the West Coast, there's been really poor identification of skates. It's been 99% of skates have been unidentified through 2010. They're starting to get a little bit better with some observer programs to start to identify skates from port sampling. There's been no baseline for the assemblage here. We don't know the species composition over time. There's been no point at which the skates were kind of surveyed and assessed for relative abundance or anything like that, so we don't have any kind of historic baseline for what's there. And in this coast, the information that we have about skates is based on anecdotal observations, usually during surveys for other ground fishes and whatnot. So not a whole lot of spatial information. In the Gulf of Alaska, we've had reliable identification back to about just before 2000. There was a real effort put into teaching fisheries observers to identify skates, and this guide was put out at that point to help out by some skate researchers at Alaska Fisheries. The assemblage in the Gulf has been characterized by the same group of researchers. That's a white blotch skate that had a range extension of about 1,000 kilometers that was caught in the eastern Gulf. They didn't know it occurred there. There's no habitat information up off of Alaska, and there's not nearly as much effort put into kind of habitat mapping and that type of thing up there. So. Okay, so my hypothesis for this section is that there are broader areas of differential importance for skates, spatial areas within their overall range, because they do overlap in depth, and that distribution abundance patterns will vary by species and life stages. There's been known segregation shown in skates between adult males and females, and between adult males and females and juveniles. And when they co-occur, they'll differ in size, is what I thought, because I figured if they're co-occurring and they're relatively the same size, these are kind of gape-limited generalist predators, then they'll compete for food. And I probably won't, and if that's the case, I won't find 2 of the same size and the same exact kind of distribution. Okay, so West Coast data collection. These are all from the National Marine Fisheries Service bottom trawl surveys of the West Coast are conducted between May and October. So this is just part of the year. They go down the coast and they go back up the coast. So we've got 357 trawls within this region. This was the region, the study region for the West Coast for the spatial portion, which is off Central California, as you can recognize. This footprint was picked because there's a complementary study at Imbari on the deep water fauna in this region, and this is kind of the footprint that we use for the Shark Center for where we're getting samples. And then we have this kind of smaller, kind of more local scale, which isn't too much smaller in this region, but where we have surveys from the National Marine Fisheries Service off Santa Cruz. The bottom trawl surveys were used for all the species except for the starry skate, which used to be called the rock skate, and we only got a couple on the trawls, but we got a ton on the long line. So model distribution for this one based on long line surveys. And we've got a whole bunch more samples in the West Coast, or in the Gulf of Alaska, much larger study site. Same thing, yearly trawls, kind of generally at the same time period that occurs up there, and you see the much larger number of trawls up there. And then we've got kind of another local scale here, which is Alaska Fish and Game Surveys conducted over the same general region, spring and summer, in these regions in the kind of extreme western Gulf. So this is where my spatial samples came from. For analysis, to answer the first couple questions, differential resort that they're using different areas in their segregation. Global and local, so I looked at a bunch of spatial statistics mostly. Global, I'm talking about the entire study site. Local is within the study site. So if you look at clustering on a global scale, then you'll see is the distribution of species clustered over the study site. Then on the local scale, you'll say like what's peak clustering occurs at what distance or something like that. You start to look at where are the highs and lows. So that's what I mean by global and local. I'm going to just present today for you one of the local statistics that's probably the most visual and easy to recognize. Hotspot analysis, essentially, neighborhood statistics. Using GetSGI as the statistics that's used, and running this in ArcMap. And I did regression models for the same questions in California, mostly zero inflated Poisson, all zero inflated Poisson models, because a lot of zero values for the skate catch, and often when you find them, you find a lot of them. Ran a bunch of different models and assessed them, but these are the ones that came out the best, and those are the variables that I had. In Alaska, there were far fewer skates, so I had to use logistic regression. And then we had habitat data for the West Coast, both from Monterey Bay, from surveys Mary was doing, and Mary Yakovich was doing. Statistics because of that. And then there's also this habitat suitability probability that's calculated from depth, latitude, and habitat preferences, and used to kind of synthesize to make a map of probability of occurrence for these species along the West Coast that Marine Fisheries uses for management. So what I did was I took their criteria for making these probability maps, and then I remade the map once I updated it all with my information, just for one skate as an example, which was a California skate. And I did that because I looked at the map and it seemed especially erroneous in any case. So spatial and size segregation, use randomization tests, and on the overall trawl data set to see if certain trawls occurred with more or less species than would be expected, and then ANOVAs and t-tests just to look at the size differences. So for the model predictions for the West Coast, I broke these into life stages whenever possible. So the J is for juveniles, T is for transitional, A is for adults. And I almost fell down. So when they don't have a life stage listed, it's everything group because we didn't have enough data for them. So basically, we can see from here is the Longnose skate was the most abundant based on the median count. But also, if you look at the common depths, they overlap considerably, right? So a lot of these are bounded by the survey, which went from 58 to roughly 55 to 60. Or 600, 55 to 600 meters. You see a lot of overlap between them, kind of a shallow. The big skate and California are generally more shallow than the Longnose and sandpaper, and a lot of overlap in the common depths. But you look at, I don't have the Oh, right. So in this region, you get basically what you look at is you look at the overall population and you run the regression models and see what the variables predict in terms of depths and median counts and temperatures and whatnot. You see a lot of overlap, but when you actually conduct the spatial analysis to look at areas of long-term aggregation, so now I'm looking at hot and cold spots. So I'm pooling all the data because we don't have enough data year by year to look this at a yearly basis, and I'm looking for basically areas where these things kind of tend to co-occur in large numbers. So the dark red is significant at less than 0.01, and the bright red is less than 0.05. So for the big skate, you can see basically where it's distributed here, northern part of Monterey Bay and up through Davenport. You're going to see that these don't overlap really at all, the areas of the primary kind of core distributions. And these starries you've got down off of the southern bay. California is more inshore in the north. And you get Longnose out on the shelf, and then sandpaper arc tilted towards the north, and there's kind of one area where they have a lot of kind of a center of population, so of aggregation. So basically what you have here is that you got long-term, so you have broad overlap if you just look at the general depths and temperatures and other tolerances, but what you see on a finer scale is that within this region, there are really these kind of core areas that distinguish them, and they're found in different places locally. This has been reported in a bunch of different areas. There's something that's being missed in this trawl analysis is that there's a lot of movements of these skates seasonally and whatnot, and some more fine scale patterns that you kind of, we're getting an overall look at where they kind of generally occur, but embedded within this is a lot of kind of variability as to how these things kind of move on a more of on a short-term basis, and often in different months that aren't represented here, because oftentimes they'll breed in the winter, and those months aren't represented in the trawls. That's when they'll tend to segregate a lot more. Okay, so I didn't find any difference between observed and expected for these. There weren't more or less skates in halls than you would expect. You would expect if they were segregating that, halls with one would be, or more common than, or you would expect that there would be halls with just like one or two skates would be significantly, would occur significantly more than predicted, and ones with more than those would be less than predicted, but I didn't see that. But what I did see is when the species co-occurred in any reasonable numbers, that they always were significantly different in size, which is one of the hypotheses I was looking at. For the habitat associations off California, we had California skate. They're not all on unsolidated bottoms, basically. California skate likes sand. Starry skates are found almost exclusively on rock reefs. Longnose skate likes mixed habitats mostly, and sandpaper skates are found on mud. So we're seeing more habitat differentiation in skates than was originally expected. And then when I reran the map for California, and one of the reasons I realized this map was wrong, right? I mean, it's like the skate hates Oregon. Like when you get right to the border, they just drop off, right? So this had to be some artifact of some kind of, grouping or something. So in any case, they had this fish down to like 1,600 meters. It was based on a misidentification. They don't get deeper than about 350. So when I updated the parameters that they were using, which a lot of them were generalized, I get this map where the center distribution for these things is really just kind of off San Francisco, which wasn't super surprising because when we would bring skates into the corporation yard from the trawl surveys, we were dorks, so we would, like Dave Ebert and Wade and I, they'd dump the bag and we'd try to guess the depth and region where it came from. And if we ever saw a bunch of California skates, we'd always say like, oh, they're off Davenport or something. And it was pretty cool that once we ran I ran all these complex models to prove what I already knew. They occur off Davenport. But in any case, now I can show Marine Fisheries, your map totally sucks, and then he can give me money to make new maps, which is kind of So, okay, model predictions. And now we're in the Gulf of Alaska assemblage. And these are the predictions of my regression models. And you see, for this, we got a little bit of the same situation where you get a bunch of overlap, especially in depth. You get the big skate up in that region is a very shallow occurring species. But a lot of overlaps in the other ones. But when you look at the areas of maximum occurrence, you start to see some serious segregation. You look at where the maximum longitude occurs, for instance, for the Alaska longnose Bering skates, which have similar depths that are really kind of far apart. And that basically plays on what I showed you earlier, or what I'm going to show you right now, actually. The spatial segregation in this area, which is also pronounced and a little bit different. It's a much larger area than we were looking at off California. The reds are significant highs, the blues are significant lows. You've got big skates centered around Kodiak, where they love to eat crabs, which we're going to talk about in a little bit, and some of the real nearshore areas. Elastic skate kind of skewed to the west, and these also kind of shallow areas. Longnose is a little bit deeper, diffuse and up north. And then you've got these two skates, the Bering and the Aleutian, that have this huge overlap here in Shelikof Strait. They're really the first two species that we've seen in either assemblage that have this broad overlap. We'll talk about that in a sec. In this region, we do have more. Oh, sorry. We have segregation by haul, and that you've got significantly less hauls with 3 or 4 species than you expect, and a lot more with one than you would expect. And then these two species that occur in Shelikof Strait, which I showed you earlier, which are the Aleutian skate and the Bering skate. It's pretty interesting. They overlap here in occurrence, but there's almost a complete separation by size. And whereas you have the full size range of the Bering skate, and then you've got this truncation. They're both born at about the same size, but you get almost very little of the Aleutian skates that are over like 800 centimeters total length in this region. And we didn't find the small Aleutian skates actually during the survey. I'm not exactly sure where they occur in any kind of numbers, but again, you have this kind of situation where when you have two species occurring together in large numbers, they tend to be a different size. So what do we learn about spatial ecology? Skate species and life stages are highly clustered in both regions, and there are long-term areas of aggregation for these species that are spatially segregated, and when they co-occur, they tend to differ in size. All right, moving on to trophic relationships. So now I've said, okay, there do seem to be some spatial differences in these species. I'm expecting to find the same type of thing, kind of, well, we've debunked kind of the idea they all occur on unconsolidated substrate, and they can kind of be generalized for distribution. Does the same thing apply to trophic relationships? Initially, I thought it probably did. So off the West Coast, for trophic ecology, we only have anecdotal information. There was a 84 thesis published by Waldo Wakefield that included some skates. And that was it until we got money to start the Shark Center. We published 4 diet studies on skates locally occurring so far from the same data set that I'm using here. And we saw variation by size, depth, sex, maturity, and season among those skates that we studied. I'm going to get into this in a second, and basically kind of how you choose your variables for diet studies and how that affects your kind of outcome and interpretation. I just think it's something that's been problematic in the field for a little while. And then in the Gulf of Alaska, there were no published diet studies and limited data from the trawl surveys. But in this region, they do food web modeling, and they've been incorporating diet data from distant regions for species that occur there, like for instance the Western Bering, when they don't have local information, and then generalizing it to that region. For instance, if they eat 80% shrimp in the Western Bering, they're picking the local shrimp in the Eastern Bering and then applying the same proportions. And I just wanted to test and see if that was a good idea or not. Guess what? It's not a good idea. Go to the Bering. Hypothesis and objectives. So I wanted to see if my hypothesis is basically that skates are trophically redundant and can be grouped or generalized. And then, so my objectives were to determine the sources and magnitude of dietary variability in each of the data sets. And then for the 2 species that co-occurred, I want to compare the trophic ecology of these 2 species to see if they're consistent or different across regions. Data collection, California, these are both, for both regions, are the more local, kind of smaller scale. These are the number of stomach samples I got from each of the different methods. Usually you don't combine long line and trawl surveys because they're different collection methods for diet. And they can bias your results, but like I said, we didn't get starries on the trawl survey, so I threw them in. I ran the model with and without the starries included for California and got real similar results. It actually wound up kicking them out of the model at the end anyway. So anyway, those are the data for Central California. And from Alaska, I had just under 12,000 samples from that kind of smaller region in the Western Gulf. So for the first objective, use percent weight data because I wanted to look at kind of energetic importance, and it's usually a better proxy than percent number for that. And then I compared a couple different, or 3 different percent weight metrics, just percent weight overall, which is just the over the entire stomach, or given a stomach sample, what is the percentage of a particular prey item in that sample, and then what is the, then I did prey weight over predator weight. So I'm actually looking at not the percentage, but the amount of the body weight that each of the prey items take up. And then I did that same metric, but scaled for differences in consumption, which kind of increases exponentially with size. And I found that they were all consistent, so I used percent weight data because it's the most kind of easily calculated and generally used. I ran a Permanova because what I wanted to do was I wanted to include the variables, all the variables I thought may be important for contributing to dietary variability based on just review of the literature, and then parse out the variability explained by each of those variables. A lot of people generally go into a diet study where they pick one or two variables to look at, and then they find that they're significantly different, but it doesn't really tell the whole story, as I'll get to in a second. And then I did dispersion analysis because a lot of times you'll compare between or among groups and you get a significant difference, but your variance between the groups might also be significantly different, which will erode your p-values. This happens a lot with really generalized predators that have really varied diets. We had a canonical correspondence analysis to basically interpret the results of my Permanova, and then use GIS to kind of explain some of the trends I saw. And then for the comparison, did basically the same thing, except I did t-tests to look at differences in trophic level between regions. So for California, I have 5 species there. So that would mean that, yeah, if we look at species differences, we have significant differences by species, very highly significant. But the species themselves are variable. The variance among the groups is variable and explains a really small kind of proportion of the variability. What we see here is basically what explains most of the variability in the data set is the haul. And that's basically the timing and location of where you fished pretty much determines what these things are going to eat. Usually what people look at are species and total length differences, which are often found to be significant, and they are here. But if you'll see, they kind of get swamped out in terms of variability explained by the timing, location of haul, which for a generalist predator, basically, so what we're starting to learn is that it seems that there's plasticity, obviously, in the spatial, the ranges of these things, depth-wise and spatially to some extent, and they probably exploit feeding opportunities like we've seen in the anecdotally in the Monterey Bay here. I haven't had enough data to really check this with or to run this with the data I have, but you'll see a lot of euphausids basically in fishes from a particular haul when they're in entrainment events or rockfish recruitment events and blah, 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 things like that. You'll see a lot of them eating the same thing. So, and then these other factors, which you say, like, you know, if you were to look at differences in maturity, you would say, yeah, they're significantly different. It doesn't mean anything, right? So I think it's really important to pick that you decide your variables rather objectively rather than subjectively for diet studies, or else you can really get a different kind of interpretation of what's going on. But basically what I saw from this, and I was surprised, only 39% of the variability explained. This is using 47 non-redundant prey categories that we made up for an essential fish habitat project for groundfish on the West Coast and applied to these with a little bit of modification that I dropped any species that didn't occur in at least 5% of the stomachs or in at least 5% of the mean weight. So in any categories, and a decent amount of variability explained, but like I said, mostly attributed to spatial and temporal differences in where you catch them. This is a hideous plot. I'm just going to walk you through a couple little things here. You got rhina, mostly rhina here, mostly rhina here. You got mostly rhina here eating shrimp and fish, mostly rhina here eating cephalopods and fish. And basically this is what you get. You get different species in different areas eating different things. And I haven't broken this out further to look at spatial temporal variability on a more kind of analytical sense. This is kind of an output from the model that is kind of a follow-up that I wanted to do, but didn't go to kind of my initial kind of question, which was what are the main source of variability. But this is something that's become somewhat interesting because everyone knew that these were generalists, but they seem to be even more extreme generalists than originally thought. So I pulled haul out of the model because it just blew up the model, which means that explains a crappy percentage of the variability, which you could kind of see from the prior graph. But I just wanted to break this down to see how things fell out here, and what you're seeing kind of underneath this Hall effect. And what you've got basically is separation by the smallest species, this sandpaper skate here, eating mostly small crustaceans, amphipods, and isopods on the first axis. And then you've got the skates here, most of the other skates, three of the other species, like I said, stars got kicked out, eating more fishes and decapod crustaceans. And then the second axis separation is you get the smaller, the juveniles and these two species eating mostly crabs, and longnose skate and skate at larger lengths and deeper are eating fishes and cephalopods. In the Gulf of Alaska, we see kind of the same general trend. That's, again, an order of magnitude more explained by Hall than by species differences. Everything else is pretty much non-significant. Well, it's significant, but it's biologically meaningless. And we get a lot of variability among the groups under comparison. In the Gulf of Alaska, you've got kind of a situation. This is what I was seeing here. Is if you were just to look at, and this is the occurrence of these different prey types in the diet of all the skates combined. So this is euphausids, and these are crabs, almost all tanner crabs. And euphausids just occurred in this one region, Shelikof Strait, in 2006. It was particularly cold, and they're really only eaten by Raja rhino, which is only the only kind of skate that was occurring at those depths in those regions. So they're opportunistically eating euphausids here. In these regions, you get Raja rhino co-occurring with big skate, and when they're co-occurring here, there's a whole bunch of tanner crabs. Everybody's eating tanner crabs. So it's just kind of a reason for pause here. If I'm just lumping these rhino data together, which often happens in diet studies, and then comparing it with big skate and not looking at kind of the spatial differences, you're going to get some proportion of the diet attributed to euphausids in longnose skate that don't occur in big skate. You're going to say, aha, like this is some source of separation. But they're not co-occurring here where they're eating where euphausids are occurring and being consumed. So it's really important to incorporate a spatial and temporal component if you're looking at dietary studies of generalist predators like skates. And again, this is kind of the same thing, what are the main source of explanation. And on our first axis here, these axes are about the same percent of variability. It's not really a species breakdown. These two species eat different things. You've got the big skate eating more fishes over here, and the Bering skate eating more of small crustaceans and invertebrates. But they've got a secondary kind of separation by depth, or you've got a separation as well by depth and total length. And on the second axis, you get some species separation where you're getting the Aleutian skate here eating some different kinds of fishes and cephalopods and these other two species. But these are all kind of underlying and embedded. Like you can see, it's only 7.5% of the variability explained. So this is what if you didn't look at Hall, this is the picture you'd get. And these are a source of variability, but they're not the main thing that's explaining diet in these species. So regional comparisons of the big skate, this is kind of interesting. Like I said, the big skates are much larger in Alaska. This is the relative size of the samples that I had. So the samples I had in Alaska were much longer. But they had significantly lower trophic levels, 3.66 to 3.86. And they ate a lot more. These are quartiles and median values. They ate a lot more fishes off of the west coast and shrimps than they do, and a lot more crabs in the Gulf of Alaska. The big skate actually is a major predator of tanner crabs in the Gulf of Alaska. About 66% of the diet was composed of tanner crabs, and they're across all sizes. Same thing with the longnose skate, same kind of relative size, not as big a difference, but larger skates up north eating lower on the trophy, eating more decapod crustaceans and less fishes. So we're seeing basically these predators serve different predatory roles in different regions, which probably have a lot to do with the forage base there. There's a regime shift in the Gulf of Alaska, and that area is dominated at this point by large decapod crustaceans, where it used to be kind of a groundfish-dominated system. There's a lot of follow-up to do here. These aren't questions that I was specifically looking at. These are kind of explanations to what I found, kind of next steps. So what I saw was co-occurring skates are trophically redundant, which I wasn't necessarily expecting. Diet composition was highly spatial and temporally variable. And what I think is that these differences, spatial temporal differences in prey abundance, create foraging opportunities for a variety of species. They're probably somewhat plastic. I know that they can, they're mostly found in kind of a broad region of occurrence. And within this region of occurrence, I think that they probably exploit foraging opportunity as they occur, and if there's a massive entrainment of something or a pulse of something, that you tend to find a lot more species eating the same things at those times. And there was extreme variation in the diet and predatory role of those two species that co-occurred. So yeah, we saw different resource use for skates. This is the end, kind of the wrap up for the first part. So one of the questions I asked is if there was differential core resource use. There was among broad overlapping tolerances, but spatial separation was a lot more pronounced than trophic separation. Spatial temporal differences in prey abundance likely influence diet. So future directions for this: incorporate seasonal data and telemetry. There's a bunch of sexual segregation that I mentioned that can't really start to look at with as well with the trawl data because a lot of these are daily movements. They've been seeing skates kind of move up off the bottom at night, make regular kind of daily migrations, also seasonal migrations. And like I said, in the winters usually when they're breeding, and that's more when they're typically more sexually segregated. I already covered the migrations. Should we be conducting diet studies on skates? You shouldn't be conducting diet studies on skates if you just want to go off the coast and pick two species and see what they're eating and if they overlap or something like that, because when you collect it is going to highly influence what you see. A lot of times skates have been used to monitor prey abundance. They've been doing this off the East Coast because they're good benthic samplers of basically what's generally available there. But that doesn't make for a very good comparison, especially if you don't have kind of a focused objectives. But if you have a hypothesis-driven study or you're looking at monitoring, then sure, trophic studies have their place. But I guess what I'm saying is there's right now a whole body of literature where if I go back and look at skate studies where they just, for instance, picked a variable like sex or maturity, and then they run a study and say that these species are significantly different, and they just give you a p-value. But I don't know what proportion of the data set is being explained by that variable versus other factors like the spatial temporal factors that I talked about here. And if it holds true from these two assemblages in other regions, then those other factors may be just kind of a minor source of variability. And one of the things I really want to look at is how can there be so many species of skates? And generally, like a Hutchinsonian view of the niche where there's competition for limiting resources usually has been supported in terrestrial systems, closed freshwater systems and things like that, and is not usually easily supported. There's very little evidence for it in open marine systems, top-down effects, food competition, things like that. And there's another theory, neutral theory, which was posited by Hubble, that biodiversity basically, he says biodiversity generally arises from a random walk, and that speciation events tend to occur in punctuated times when niches open up and things like that. And it's interesting because they've just recently done they've done phylogeny of about half of all the existing species of chondrichthyans. And the skates show one huge radiation based on how they've kind of mapped that back in geologic time. Anyway, this is something I'm exploring a little further with some other ecologists up the coast. Okay, so I wanted to apply my ecological studies to help improve skate management in the two regions. Like I said, based on ecosystem approaches, not there are traditional stock assessments that occur in these regions, but this is more using ecological data to inform management. So on the West Coast, there's only one species that's currently in the management plan or is managed with the quota. That's the longnose skate. And these other species that I mentioned, my four other species, have kind of been moved in and out of management. And right now there's concern that the big skate should be managed because it's been declared overfished off of the Northwest for the last couple of years. And things are a bit in flux right now. And actually, skates are starting to get some management attention. There's interest in doing stock assessments. There's only one stock assessment right now for the longnose, and it's not really valued very highly. And there's a kind of push to do stock assessments for big and California skates. The other thing is off the West Coast, there's been a lot of spatial management of groundfishes through MPAs, no-take areas, that type of thing. So a lot of the management in this coast or the ecological or ecosystem approaches to management have been based on more of the allocation of space than food, and then kind of the separation of food or the distribution of, or the use of food resources, food web modeling, that type of thing. And these are kind of managed through those HSP maps that I showed you, the habitat suitability probability use maps, and there's a database basically that's collected for these ground fishes that goes into informing management, not as directly and specifically as it does off Alaska, but they're starting to incorporate some of these ideas. So for the West Coast, and I broke these down by regions for this part of it because they're really distinct as to what they're doing. I wanted to estimate the distribution abundance patterns of skate species and life stages, and then the relative importance of these three factors. Right now, they're treated equally in the habitat suitability probability models, but species life stage. So for the first two, I used the same trawl data set from 2003 to 2013. I ran a GAM to get the depth temperature relationship, and then ZIP and zero inflative negative binomial models to model the distributions. I wanted to basically get a predicted model of the distribution that then could apply back to kind of make like a Kriegen or some kind of a service like that, where I could use areas where they didn't survey and kind of predict what you would find in those regions, and AIC for model selection. And then I got the absolute and preferred, absolute ranges of depth, temperature, those type of things. The preferred was the common ranges. I refer to the common ranges as areas where my predicted Oh, okay, so basically what you see is, and this happens regularly, you've got counts of up to 49. So what you get is a ton of zero counts, right? These are quartiles. You get a bunch of zero counts, and then in the last quartile, I get from 1 to 49. So basically, you're getting a lot of zero values, and then a couple large values. And what the model predicts is it tends to underpredict the large values and overpredict the numbers of values that have, or the number of hauls that have actual positive occurrence. It's just a function of how the distribution of these, what the distribution of these fishes are like, because they tend to be like, I guess what we've seen from the spatial results and some of the things I've been saying, they kind of, you'll tend to find large aggregations in particular areas at particular times. And for this species, depth was more important than latitude, which is more important than habitat. Also, I'm sorry, this is for the juveniles, and then fewer adults. Same basic take home is that it kind of underpredicts the maximums, overpredicts some of the, but these are the best models we have now. They didn't have the substrate information completed for this, and habitat was, for this species, not super important, but some of them it was. So this is part of kind of an ongoing work I'm doing to kind of build integrated models for these skates with personnel at marine fisheries. In the Gulf of Alaska, they use a food web model to inform stock assessment through energy flows. Longnose skate distinctly, and then they lump all the soft-nose skate, the Bathyraja species. They do stock assessments for the skates every year, and they, like I said, they have this ecosystem considerations section that helps inform management up there. And food web modeling is the main source of kind of an ecological ecosystem approach up there. So let's see, my objectives were to characterize the spatial, temporal, and length-based diet of these, of the skates based on my trophic results, and use that to estimate consumption on a per capita basis and total removals, and then update the Gulf of Alaska food web model for skates. So for objective 1, I summarized the diet by region, year, and size class. And then to estimate consumption, this is a gastric evacuation model. You just need to know this is the mean temperature, this is the mean weight of stomach contents. These two are constants for elasmobranchs that are typically used. And you only had data, enough data to model this in the central region, and had to extrapolate to the other regions based on kind of relative abundance. And then updated the food web based on what I found from these two. Went back and updated the food web model, but with new diet composition and consumption estimates, and then reran the model to see how it differed from the original model. Original model was built in the early 1990s, and the new model, all I'm changing, and the only thing, the model was static. I changed the skate inputs, I should say that, because in both these regions, I'm doing things that are a little bit ahead of where they were at the time. Like, for instance, they now have updated this whole food web model, whereas I can actually compare the skates at the same time period with the teleosts at the same time period. But for what I did here is the only inputs I could change were the skate inputs. So in any case, what I found is tanner crab consumption was greatest around Kodiak Island. The skates up in Alaska eat a ton of tanner crabs. Euphausias were more important. I'm sorry, I think I said 2006. In 2007, Shellicott Strait, when it was that cold water year. Amphipods and polychaetes are only eaten by Bering skate, and this is consistent. We always see basically in any area you get small skates like under 50 centimeters or something tend to eat amphipods and polychaetes. This general kind of truism in that you get a little bit bigger, they move to decapods, and a little bit of fish isn't bigger and more piscivorous. This seems to occur universally among skates and within a species differentially in different regions, like I showed for big longnose skate. You're still getting increased piscivority in the Gulf of Alaska with size, but the overall piscivority in the Gulf of Alaska, for instance, is much less than it is off California. Consumption comparisons. Earlier I said that with metabolism you see, with increasing size, increasing metabolic demand, you see a lot more consumption. These skates are those that are less than 50 centimeters, and you see this as a grams of weight per day that's estimated. Medians, first and third quartiles, and min and max values. And then what comes out of these plots basically is at the same size, big skates generally tend to have higher consumption rates than the other skates do. A lot of variability here. And they tend to be more shallower occurring. So here this species has and these are, like I said, these are median quartiles or whatever. These significant results actually refer to like a comparison of means between the three of them. So these data are not completely consistent, but they're not directly consistent between what I'm showing here and what these values are. There's a difference in the means. You can see like the elutions are consistently smaller than or lower consumption rates than most, except the longnose. In this year, longnose and big second, and then these big skates in 2007 consume the most. Big skates are the largest of these, and they also occur in the shallowest region. So it's not too surprising. But before they had consumption rates just grouped by overall for skates. So this allows us to start breaking them down by species for the models. One of the interesting things here is consumptive removals, and this is throughout the entire Gulf. What you see tons of tanner crab consumption by big skates, and that for all skates, they consume many more metric tons of pandalid shrimps and tanner crabs than the fisheries do. There's almost been no fishery for tanner crabs in the Gulf of Alaska for a long time, and it hasn't recovered. Although there seem to be a ton of tanner crabs around based on the stomach contents of the big skates, so this is kind of an interesting result. And the fishes, they eat just a tiny proportion of the fishes up there compared to what the fishery takes. And you get the same kind of situation in 2007, except that you get a lot more tanner crab consumption in 2007. So you can see between years, even these things diets are somewhat variable, as I've been kind of saying throughout. So for the food web models, we've got an overall increase in skate biomass between these time periods. That's the percentage increase for each of them. Change the consumption of biomass rates, which were flat at 2 for each of them, which seemed like not a bad estimate for lazabranchs, given that kind of my data spread among these around 2. And this is the largest shallowish species, and these species are found in deeper waters. So that wasn't really, that was a nice outcome and not kind of unexpected, I guess, but helped improve the model inputs. My differences in diet from what I entered from the Gulf compared to what was there before, which was mostly from the from the Gulf of Alaska, which was mostly from the cold from the Western Bering, were wildly different. So again, hard to track because I've got differences in between regions and between years. I didn't have data in the Gulf from the prior modeling effort. Those were all pulled from somewhere else. But you can see that the inputs to the model were really different than what we're seeing right now. So they're not going to be predicting the proper outputs. What you see from my updated model is that this is the old model, the percent flow, and this is the predatory impact of skates on tanner crabs in the old model, and this is relative to the new model. So, like I said, if you update these models for other species, I'm assuming that given that there's a regime change and whatnot, I'm sure that the impact of skates will be mitigated. But this is kind of a first step to see what impact might skates have, and then actually following up on making these model updates, like I mentioned. They've been updated for the other species now. the study, and they're found to be major predators of tanner crabs. Future directions. Now that we have a substrate map that was from some work that was completed as part of the last Central Fish Habitat Review that it can then use with the models I've created to basically do predictive services through kriging and update some probability maps for the coast that way. And then starting to incorporate trophic information. There's been starting to be some Atlantis and ecosystem models used on this West Coast to look at trade-offs and other things in fisheries. We're starting to feed in some of the trophic information into the habitat use database that's being revised so it could be used for stock assessment. So there is some movement towards starting to incorporate trophic information on the West Coast from skates and from other ground fishes. Gulf of Alaska, working to update the overall food web model with skates and with the other fishes so we can see what the impact is now in a direct comparison. And starting to do the same kind of spatial modeling that I was doing on the West Coast with the Gulf, except we don't have any habitat information up there, just depth and latitude. So the conclusions, people should learn how to identify skates. It's a hassle trying to go back through records that are unidentified skate and make sense of anything, especially now that people seem to have an interest in, all of a sudden now there's an interest in skates and what their impact might be and what their populations might be like. We really are having a tough time sorting that out because we're establishing kind of baselines right now. Skates all look the same to some people or to most people, and they're not extremely charismatic. I mean, I get that. But they're not that hard to identify, really, because you can use meuristics, thorn counts, and other things to separate them. And even if you can't separate all the species in the area, it's really easy to separate at least a few of them and maybe lump a couple so that, and even that does a whole lot when you're trying to look back and assess the population. Stop conducting diet studies on skates if you just want to figure out what they eat, you know, and do these two things differ by total length or something like that? They're extreme generalists, and where and when you fish is going to largely dictate this. So unless you account for this and have a kind of a hypothesis that accounts for the fact you're going to get extreme variability, which means, you know, how you're going to sample and the number of samples you're going to take is going to be hugely important. These kind of willy-nilly studies where you're just picking a variable and looking to see if it's significantly different between two groups that may have been taken at different times in different places doesn't really tell you anything. And I think research efforts should be more focused on fine scale and long-term spatial studies. I think it ties together the food and space utilization, where they're going to forage, things like that, where they go on breeding migrations, how they segregate in more of a finer scale, are things that you can really only learn, not so much from the trawl studies, but from actually like tagging and tracking studies. And then one of the interesting things I saw is that core resource use really varies between skates and rays. Rays have a variety of modifications for feeding. They can filter feed. You've got electric rays that shock things. You've got rays that, durophagous rays that crush. You've got rays with teeth that look like white sharks if you look at them under a microscope that eat fish. You've got ones with circumscribed mouths that suck polychaetes out of burrows. You have all these feeding adaptations. In skates, none of them can excavate a feeding pit. They can't crush anything. They are basically gape-limited predators, and they do have something cool. They have chitinase in their stomachs. They eat decapods whole, and they just slowly dissolve, which is kind of cool. Some of the other rays just kind of have to crush them up and stuff. But what you see for the rays is you'll see tons of them occurring in an embayment that's maybe 5 meters deep. You'll see 8 or 9 species. Their spatial overlap is almost like complete in these little regions. And you'll have some that excavate feeding pits. You have some that, and the food overlap in these regions often is really minimal. So it looks like there's difference in core resource use, at least in the groups, in the assemblages I looked at and the ray assemblages I know, between space and skates and food and rays. And with that, I'd like to acknowledge a bunch of people you don't know from University of Washington, and Dave Ebert. Jason Cope, you remember, right? Jason Cope is one of my best friends, and he's a genius. And all the PSRC students, many of whom collected data that I used for this project. And those are my funding sources. And I want to thank you all for coming today. And I guess we have time because there's no one else and no one's going anywhere. You feel free to ask me some questions. I would love to entertain them. Thanks.
So, Joe, I really appreciate your attention some of the stats, it seems to be that you've kind of got competing ideas here in terms of generalist predators and spatial separation. Because if they're really that generalist, why is there spatial separation?
It's a great question.
So what I'm wondering is if it's possible that they are actually more specialized, but by length or by age or or by season or something. And I mean, you touched upon that in terms of the understanding the movements. But what do you think about that kind of alternative hypothesis that they are more specialized, but by life stage or something?
I definitely think there might be a component, an unaccounted for component that could explain more of a difference in dietary variability than I'm seeing here. So yeah, size of prey, maybe more lower taxonomic distinctions, although I couldn't really get at that with the data. But there are strange. Okay, so there are a bunch of skates have a bunch of strange things that I think keep, in terms of like species recognition, and they have electrical organs in their tails that are only that are used for kind of conspecific mate recognition and whatnot. They have these other kind of behavioral mechanisms that go into their kind of spatial associations and movement patterns that I think also are probably missed. And so I don't know how widespread, I don't know how much more trophically distinct they're going to be in the same region. I think that then in the same regions that they generally do overlap, because I got such strong differences with a pretty good number, a large number of different prey categories in the same regions. But there's definitely something else going on here. And I don't think that they subscribe to kind of a normal question, Rick. I'm hoping to explore this kind of in more detail. I wasn't really looking at, my initial question wasn't really why there are so many skates. It was to look at these kind of two aspects of the niche and see if they were really, if they really held up based on what we've seen in literature. And I don't think space did. Diet seemed to, but yeah, there could certainly be some underlying factor that I haven't accounted for there. I don't know. There's 300 species of these things. Their feeding mechanism is really conservative throughout. I don't know. Anybody has some ideas, I would love to talk about it. I'm starting just to kick these ideas around with more people that do kind of evolutionary process and stuff, which isn't really what I typically do, but it's a very interesting question to me. Kind of just starting to get at it. Yeah.
So Joe, where's the center of their evolutionary history? Where they begin, do we know?
Yeah, at the sea. So I don't know where that was in terms of when everything was kind of squished together in terms of geologic history, or where that opened up. That was kind of where their first radiation is, and then from kind of the poles down, I guess.
And so how does North Pacific compare to the rest of the world as far as skate diversity?
Alaska is extremely diverse. California, West Coast is not very diverse. At the depths of 1,000 meters and shallower. Alaska is rather diverse. South Africa is extremely diverse. West Coast of our West Coast is not very diverse. Northeast is extremely diverse. There's some real
Diversity.
you're talking in terms of food.
Yeah.
But what if food doesn't matter? They just kind of eat whatever fish and crustacean happens to be in
So
their
that
region.
is my next question. Right.
Yeah.
Yeah, one more.
Yeah.
We're all stopped. So I'm fascinated about the 1,000 one. So are they coming off the bottom, eat those guys?
No, apparently, they do come off the bottom. They come off the bottom at night, mostly larger specimens, so skates about 80 centimeters to a meter, and they usually come up off the bottom to hunt forage fishes and other things, because the idea is at that point, they feel like they're big enough that they can come off the bottom and not have it incur a bunch of mortality. Also at night, they have senses that teleosts don't have, electroreception, mostly, that they can use to hunt these things in the dark and eat them. The euphausids are mostly eaten by juveniles. They'll be eaten by adults and whatnot when they seem to occur in huge numbers. When everybody's eating them, you'll see the larger ones eat them. But usually what happens, it's been reported in Monterey Bay here a bunch of times, is euphausids will vertically migrate and then get infected towards shore and trained in the heads of canyons. And so we'll find hauls for a particular area, and they're usually actually from our data in the canyon heads, but not always. Oftentimes out on the outer shelf, so maybe not in the canyon heads, but at like 160 meters, 180 meters. And that's an area where you have a whole bunch of skates co-occurring. Every species that I presented here could be found, if you pulled a haul at 200 meters, you can get any of them. And when we get euphausid events like that, and there was also a blue rockfish entrainment event, you tend to find a lot of species in the haul, and they're all eating euphausids. Like I said, I don't have enough data on that, so I tried to work it up in some kind of quantitative way, but I really couldn't do it. But to me, it looks like when there's a huge pulse of prey, that you've got a bunch of skates kind of focusing on different species. And if you looked at that same area at some other time and you trawled it, you would get kind of a different species composition, which would mostly be the thing that's most abundant at that depth, generally.
Do you think the Yankees are going to make the playoff?
No, I like Lou Severino. I think he may have a good year. There's one of the best proposals, one of the most hilarious proposals I've ever heard was from fishermen on the northeast coast, because they fish down the cod. You probably know this, even though you're not. Yeah. So they fish down, and forever they've been trying to say, like, why are they not cod not coming back? They had to kind of come back, and cod really haven't. And for a while they were blaming elasmobranch. There was this elasmobranch outburst, they said, and then they really studied it, and it really didn't have anything to do with that. It wasn't, it wasn't based on kind of food competition or whatever. So what are they doing with this? What does it mean? So what they're doing is they're trying to figure out, okay, out of the species, the common species in this area, we've got a general abundance for them, we've got a consumption rate, this is how much prey they remove from that. And basically what they're doing is, okay, if we have these food web model outputs for all these different groundfish, how many tanner crabs are they eating? Okay, well, if the groundfish need to take this much, and then we can't take, we're gonna offset the quota that we would have established versus what we need to save for the fishes, because we have this idea of kind of energy flow. And this one species is super important to the food web, so we can't fish as much of it as we want. That's how they use the food web
Those
model.
models are regulating the fishers take based on what kind of animal.
Right, they'll set a stock assessment from the quota, which is how much they think they can take, and then they'll reduce it based on, if they think they need to reduce it, based on how much forage they think they need to leave for the other species. Yeah. But if they have a crappy model that's pulled from Russian data, it doesn't really work. One of the things I loved about doing this is because for my last chapter, I wanted to do things that they hadn't done yet. So I was like, I'd read things from 2005 saying like, oh, we're going to do this, and then it would be like 2015, and it still wasn't done. So I was like, well, give me the data you have. I'll do what I can with it. And then they were like, what? And all of a sudden they're like, they're eating that many tanner crabs. Now, like, they care. They're like, okay, well, let's fix them all. Because holy shit, we haven't been able to fish tanner crabs here for like 20 years. Maybe the skates are eating all that. I'm like, you know, so it's kind of cool watching like this like create interest. Because initially they were like, you know,